Welcome back. We are talking about mitosis and meiosis, and so we did mitosis, and we are halfway through meiosis. So we did meiosis 1, and now we're into meiosis 2. So let's just recap meiosis 1 very, very quickly. So we started off with a diploid cell, and the cell did DNA replication, and that gave us each chromosome replicated to form a pair of sister chromatids. And then when we went into meiosis 1, the pairs of sister chromatids called the homologous chromosomes, so chromosome 1 from mom and 1 from dad, they paired up, and then we had the crossing over event. That's the first place we got some genetic diversity. And then we had metaphase 1, where the homologous pairs lined up in the middle of the cell. Remember, they could line up, mom on this side, dad on this side, or the other way around, and that was independent assortment, and that gave us more genetic variation. And then the end of meiosis 1, the homologous chromosomes were separated, and then the cell divided, and we had two cells, and each cell only had one set of chromosomes, and so each cell was haploid. So we're now into meiosis 2, and so let's start off looking at meiosis 2, and we're going to take a look at each of those two cells that we produced from meiosis 1. So meiosis 2, we're going to start in prophase 2. We're going to follow the nuclear envelope and watch that break down. The centrosomes, we're going to watch their behavior, but most importantly, it's the behavior of the chromosomes. So remember, over here on the left, top left here, is what we're seeing is chromosome 1 from dad, chromosome 2 from dad, with a little bit from chromosome 2 from mom due to the crossing over in prophase 1, and then chromosome 3 from mom, and that's got a little bit of blue from dad due to the crossing over. And the other event, the other cell down here, will basically be the reciprocal of that. So 1 from mom, 2 from mom largely, and 3 from dad with a little bit from mom. So those are the two cells produced by meiosis 1. Let's follow just one cell through, and you can then look at the second cell in your own time. Let's follow the top cell. So first thing, prophase 1. Nucleus is intact, chromosomes condense down, and the centrosomes start to separate. As we then go into prometaphase 2, the centrosomes are completely separated, the spindle starts to form, those microtubules start to radiate out from the centrosomes. They grab, the microtubules grab the kinetochore proteins that are on the centromeres of the pairs of sister chromatids, and then the chromosomes are going to line up in metaphase 2. So they're pushed around, they line up in metaphase 2. Okay, so we've got chromosome 1 at the bottom, in the middle here, 2 at the bottom, 3 at the top. They could be in any order, it's not important. So let's see what happens as we go from metaphase into anaphase 2. So is what we're going to look at now is the spindle is going to pull the two chromatids apart. So the two sisters are going to separate. So here's the key difference between meiosis 1 and meiosis 2. Meiosis 1 separates the homologs, and meiosis 2 separates the sister chromatids. So this process is more like mitosis than it is meiosis 1, really. So meiosis 2 is more like mitosis, and meiosis 1 is kind of a little bit different. So think about the similarities between meiosis 2 and mitosis. And so once we've done anaphase and we've separated those sisters out and the cells divide, each of the two cells divides in half, and that's going to give us 4 haploid cells. So we started off with a diploid cell, and we went through a round of DNA replication, and we separated the homologs out, and then we separated the sisters out. And so each of the new cells, which will go on to be a sperm or an egg, is going to be haploid and have only one set of chromosomes. So that's one chromosome 1, 2, 3, 4, all the way up for you and I to one X or a Y. Okay, you're going to have both. You can't do an arrow, but you have an X or a Y. So let's do a couple of questions really quickly and see how this is looking for you. Okay, so let's just do a quick recap of this process. It's a real quick overview. So we started out with a cell prior to going into meiosis. So it's in G1, so that's this G1 phase, and we've got a pair of homologous chromosomes here. This is chromosome 1 that's from mom and chromosome 1 that's from dad. They came in from each parent at the point of fertilization. And so we go through S phase, and so that means we replicate each of those two pieces of DNA. So there's now 2 pieces from mom and 2 pieces from dad, and they form a homologous pair. They get together in prophase of meiosis 1, and there's some crossing over, there's some exchange of genetic information. We're not showing that here for simplicity. Meiosis 1, we basically take the homologs and we separate them. And we divide those cells to give us 2 cells, And then we have meiosis 2, where we take the sisters and separate the sisters. Okay, so meiosis 1 separates the homologous chromosome, meiosis 2 separates the sisters. Okay, so that's a real quick overview of the whole process when we think about everything taken together. Okay, so that wraps this part up. Now we've done mitosis and meiosis 1 and meiosis 2. I'll see you next time.